Okay, so the subtitle for my talk is really the unreasonable effectiveness of linear methods. I want to convince you that although the brain is nonlinear, of course the neurons are nonlinear, the synapses are nonlinear, we can get a lot of information by starting with applying linear methods and see how far we can push them and see what we can learn from them. And then, of course, when they break, because they will eventually break, the way in which they break, I think, can be used to gain some intuition about how to go nonlinear. Because once you open the door to nonlinear methods, there are so many different possibilities that sometimes it's hard to get an intuition of how to go nonlinear, what kind of nonlinear approach to use. So start linear, push it until it breaks, and that will give you some intuition for nonlinear methods. That's the main message for my talk. So now, of course, I don't know how to make anything work here. How do I go to the next transparency? No. That's
the space bar.
Next one?
The space bar.
Oh, the space bar.
Yeah.
It was simpler than I was thinking. So I have collaborated for many years with my colleague Lee Miller at Northwestern. He runs a monkey lab, and he is very interested in motor control, in how the premotor cortex and the motor cortex, the primary motor cortex M1, control movement. And control movement in this case means controlling the activity of muscles with themselves, then control the joint angles that allow your limbs to move. And again, when I first joined this lab, they were using decoders from neural activity to make predictions about kinematics or about muscle activation. And all their predictors were linear predictors. And I thought they were totally crazy. Again, I said, you know, you have this very nonlinear brain, and why are you using linear predictors? And they said, because it works. And that was very eye-opening to me, because I said, well, I need to understand why it works to use a linear predictor. So let me talk about this very simple task that I'll use a couple of times as an example. It's what is called a center-out task. The monkey or the human, we also did do this with non-human primates and human primates. You start at the center is surrounded by 8 targets organized in a circle. One of them flashes. You have to wait for a go signal. That's a variable period in trial to trial between half a second and a full second. When you get the go signal, you do a movement, in this case with a lever that controls the cursor in a screen, makes a movement to the target, you come back, you get rewarded. So it's a very simple task, and now I need to go back. Okay, so we implant electrode arrays in primary motor cortex, in the contralateral area, in the arm area of primary motor cortex, and we get recordings like this. These electrode arrays allow us to record of the order of 100 neurons. If you use Neuralinks, you can record of the order of 1,000 neurons, a few hundred, if not 1,000. And here I'm showing you spikes. Every row is a column, and I'm showing you about 20 seconds of spikes. This was actually for a human subject that was doing exactly the same center-out task while we were waiting for epilepsy-related surgery. It was a very small clinical trial. So, you know, we are theorists. How do we analyze the population activity? We take time. We bin time in bins of size delta. So instead of thinking of time as a continuous index, I think of time as a discrete index that counts the bins. And for every neuron I going from 1 to n, I measure n sub I of k, the number of spikes emitted by neuron I in bin number k. And I arrange all this data in a data matrix that has the geometry of the raster, in the sense that every row is a neuron and every column is a time bin. And I put these variables n sub I of k, activity or number of spikes emitted by neuron I and bin k. So the row has T columns, where T is this discrete time index, is the number of bins related to the duration of the experiment, and has D rows, where D is what people call the ambient dimension, is the number of neurons that you're recording from. Now, the number of neurons modulated by even a simple reach is enormous. Even a simple arm reach modulates of the order of 1 million neurons in your primary motor cortex. And as glad as we are that we are recording a few hundred neurons simultaneously, we must always remember that we're extremely subsampling the population that is responsible for producing the motion. So the simplest decoding problem would be, I show you again this raster, the monkey is making movements to these 8 targets, I've color coded the targets. I'm showing you an interval in which there have been 3 reaches, one for the yellow target, one for the red target, okay, you know what I mean, here, yellow target, red target, blue target. And so I can put a very, just to start very simply, instead of following the trajectories, I can put a very large pin, in this case 350 milliseconds, and count the number of spikes, and I put it just when they go at the go time, so that it's not contaminated by preparation into movement, but by the movement itself. And then I go to a space where every axis represents the activity of a neuron. That's the neural space, the empirical neural space. These are the neurons that I measure, and I put a point. And when I put the point, I get a royal bends, because I get points after I put every point for each trial, and I color them according to their targets. Every point is surrounded by points of any other color. So it seems that the location of the point is not very indicative of what was the trial, the target on that particular trial. But the problem is that you cannot do a 3-dimensional projection of an 8-dimensional space by picking 3 neurons at random, as I did here. You have to do it in a somewhat more intelligent way, guided by the activity itself, guided by the population activity. And this is what was done in this very, very important paper, which is now very old, you know, it's like more than 15 years old, that came from the lab of Krishna Shenoy at Stanford, where they used this axis called neural modes. Now, a neural mode is, I started with a space where every axis was a neuron. The neural modes are a direction in that space that combine neurons with particular weights in a particular proportion that is trying to reflect the covariation of those neurons. So they found a think principal component analysis. This was a little bit more sophisticated, was factors analysis, but it's just a variant of principal component analysis. So you go to PC1, PC2, PC3 space, you put all your points there, and again, after you're done putting all your points, you color them. And when you color them, you see this beautiful structure. There is a well-defined clustering having to do with each one of the targets, and moreover, the order of the clusters, if you see here, the order of the clusters goes from red to orange to yellow to green, reflects the order of the targets in real space. So this very abstract representation in which took data, we computed the factors, we from outside plotted in this particular way, shows us that the neural representation is capturing something very, very simple about the organization of the task, that is 2-dimensional, 3-dimensional targets that are actually organized in the same way that they are in real space. So what is interesting about this plot that I'm showing you here is, again, I have a very large population of neurons. I put 3 electrodes into it. The neural space is N1, N2, and 3, the activity of these 3 neurons. Every time that I put a pin somewhere, I get a point in that space. And this observation that Cunningham and you made also many years ago is that when you follow these trajectories, of course the trajectory is on field space, but they are confined to a regional space, states that are visited by the system. But you could have expected that in that small region, they fill the space. They subtend some kind of a cigar that has the same dimensionality of the full space. But that's not what we see. There is a lot of empirical evidence that these trajectories lead in a manifold that could be flat, a hyperplane, or could be a bit curved, but this manifold has a dimensionality which is much smaller than the dimensionality of the neural space. So we have this picture then that we have to do dimensionality reduction. If you do something like PCA, you will get a hyperplane. That's very, very easy to discuss. When you get a hyperplane, you get these 2 eigenvectors, U1 and U2. They are a unit length, perpendicular to each other, and they give you a basis. Every time in the hyperplane can be described by saying what are the coordinates along U1 and the coordinates along U2. More typically, you have a curved manifold, and then again, locally, you can define the bases, but these bases will move around as you move in the manifold. So we have to work a little bit harder and learn from colleagues who know about how to do differential geometry and topological data analysis in order to describe these manifolds. I want to tell you two things about this and then show you a very useful example. Let's start with the case of hyperplanes so that we can be simple. So again, I can project this trajectory in the direction of U1 and project it in the direction of U2. And I get a one-dimensional dynamics that I call L1 and L2. Those are the latent variables that you hear so much about. The latent variables are the projection of the neural trajectory along the neural modes, along these special directions in space. And the other important thing to know about is that these latent variables, L1 and L2, are a generative model. From these variables, I could reconstruct the neural activity. So these are really the building blocks of the population activity. I don't need to follow every one of the neurons independently. If I follow this much smaller number of degrees of freedom, which are the latent variables, I can reconstruct the neural activity. So that's very promising, but the question is, are these latent variables informative? And by informative, I mean, do they allow me to make predictions about behavior, be it the activation of the muscles or the movement of the limb in space? So we again went back to this center-out task, and we noticed that the monkey was very, very stereotypical. You know, this is a monkey that has been doing this task for more than 2 years. And you can see that the trajectories are very stereotypical. And if you pick one of the directions up and to the right, and you look at the X and Y profiles of the velocity, they are also very highly correlated. You can see here that the correlations are very close to 1 when we gather all the data. So we made an hypothesis. We said, look, if the behavior is so stable, there has to be neural activity that is very stable. But how do we find it? You know, the electrode moves, it picks up neurons that it wasn't picking up before, scar tissue forms, and then you lose some neurons that you were recording. So if I look at day 1 and I look at day n, I'm not recording from the same neurons. So how am I going to compare neural activity to see is stable or not? And this is where we had our first, at least for me, my first illumination on the power of linear methods. I said, okay, in the brain, there is a real neural space with a mother manifold with a real trajectory, very high dimensional. I'm not observing it. I only have access to the ambient dimension, to the neurons I'm recording from. So on day 1, I'm recording from these 3 neurons that I call N1, N2, and 3. When I come back on day n, I'm recording maybe some are the same, but some are different. I call it N4, N5, and 6. That projection is a linear projection from the true neural space to the empirical neural space. Now, every day that I do this in the empirical neural space, I'm going to do a PCA analysis, which will give me a linear projection into a low-dimensional manifold that is flat, is a hyperplane. And then I get these trajectories that look very different from each other. I'll show you here. I get these very different trajectories, this and this, and I ask myself, is there a linear transformation of these 2 spaces that will allow me to align the trajectories? And the answer is yes. We learned this method from Bach and Jordan. It's a beautiful method. It just allows you, if your manifolds are for dimension D, it allows you to find 2 matrices of dimension D by D. And these matrices come from doing some kind of, QR decomposition followed by a singular value decomposition. So again, those are all linear algebra techniques that we learn and we know how to do. And once you have your matrix M and N, on day M and on day N, you apply them to latent variables and you transform the latent variables linearly. And when you do that, we expected to be able to align them, and our expectations were much more than what we expected. I mean, what happened was much more than our expectations. So here I'm showing you a monkey day 1, day 32. I'm only showing you 8 trajectories. I'm averaging all the trajectories to the yellow target, all the trajectories to the red target, to the blue target. So I'm showing you these 8 trajectories. This is the neural space in day 1. neurospace in day 32, those trajectories don't look at all like each other. I run my canonical correlation transformation and I transform day 1 onto this and day 32 onto that. And now you see that something amazing happened. These overlap, they show the same structure. They reflect the structure of the dust because it's a petal. I go out and I come back. I'm now looking at a 5 millisecond resolution. So I can really track the trajectory. And moreover, if you look again at the color code, you will see that these petals are organized in the same way that the targets are organized in the external space. So doing this alignment allowed me to capture something about the true manifold that I could not see by looking at any one of the individual projections. I couldn't see it on day 1 and I couldn't see it on day 32. But by combining these representations and looking for something in common, I was able to unveil this. And I want to show you another, well, we have done a lot of decoding, as I said, where we try to decode M1 activity into muscle activity. And when we do that, again, we use linear decoders, and we ask ourselves, okay, I know how well I can decode with all the neurons. Are these latent variables informative? And we see that they are. And what I'm showing you here is the black curve is a typical PCA plot, is the variance accounted for. And as you see, it grows. I have a lot of small eigenvalues. This grows very slowly. And if I ask, like typically say, people say, I want to capture 90% of the variance, I would need of the order of 40 components to do that. But now I use the mirror lab decoders to predict muscle activity. It's not a very good prediction. These muscles are measured externally. They are not needles, they are bands. So it's a noisy measure. And then other predictors are linear. So it's not a great prediction. So these circles at the end, this is a collection of muscles. Some of them are flexors, some of them are extensors. But it just shows you how well I can do to predict the envelope of the activity of that muscle using all the neurons. So now I go back and I say, well, let me construct a predictor based on the L1, and then L1 plus L2, and then L1 plus L2 plus L3. And as you add more latent variables, you do better, but notice how quickly you achieve the same result as you had achieved if you had used all neurons. So just a little, by the time you have 10, you are doing as well as you did with all the neurons. So this says that if I keep a 10-dimensional PCA space, I'm capturing enough information about neural activity from the point of view of the neural activity allowing me to predict muscle contraction, muscle activation. Of course, a lot more is happening in M1 because you can see that the variance accounted for continues to grow and grow and grow, right? And however, we can use, if we do the dimensionality reduction in a smart way, we can find latent variables that are informative for the task at hand. And let me show you one more thing. The other thing that we ask ourselves is what happens if I compare tasks? So here is a monkey doing again central tasks, that 2 dimensions or 1 dimension, and then doing tasks that are completely unrelated to reaches, like putting a ball in a tube and picking it up at the end, or aligning with a tilted representation. We can do, again, the variance accounted for and find the manifolds. We find, again, 80% in this case. By the time we keep 10, 15 dimensions, we capture 80%. And we have done it for the reaching out tasks and for the ball and tube tasks that are very, very different. And now, again, we go linear. We said, okay, how do I know if these manifolds are very different? For instance, all the reach tasks maybe are more similar to each other than the ball task. Do the manifolds show me that? And then again, we went linear. We can compute principal angles between the manifolds. And what we see again is that all these principal angles, when you do them pairwise, comparing 2 of the rich tasks, they are very small. This is a control. So these angles are really, there is a whole collection of very small angles. And again, when you compare the ball versus tube task, you have at least five dimensions along which these angles are very small. And this is interesting because it indicates learning facilitation. If you have learned, first of all, we learned that the manifolds are task specific, but at the same time, if you have learned a task and now you learn a task that has some similarities with it, some of the neural modes are oriented almost in the same direction, you can use them, and you have only to learn the additional neural modes that point in very different directions. So this indicates facilitation in learning, and there are some beautiful experiments done by my colleague Aaron Batista at Pittsburgh that show that. So this is the summary of what I wanted to tell you. We now do experiments of behaving in a rich environment and all sorts of behaviors, picking trees, hanging from the ceiling, scratching your back, to make sure that this low dimensionality was not an artifact of the simplicity of the task. We try to do tasks that are a bit closer to real behavior. And these are the people I collaborated with, and thank you.
There's time for two quick questions.
Well, this very nice, and I think this question you raised about linearity, because sometimes you see neurons that actually integrate information in a linear way, even though the default, if you choose it, is non-linear. So, so maybe it is that linearity is good for some kind of signal fidelity, but for the decision is non-linear. But maybe the brain likes to be linear as much as it can, and then make a non-linearity in the end to make a decision.
So I'll tell you my hypothesis about this, but it's an hypothesis, it's not something that I can show. I think the brain linearizes. When you start, for instance, with V1, V1 is representing the world. And the most important thing in the world are the edges. If something makes a caricature of you with only edges, people will recognize. It doesn't matter how the luminance changes as you go smoothly down your cheek. The edges matter. The edges are non-linear, and I expect V1 to create a very non-linear representation of the objects of the world. But I think as you go from sensory to motor, the system gradually linearizes. And by the time you get to motor cortex, you have these, with the manifolds that we find are a little bit wiggly, but they are not Swiss rolls. They are just, not exactly flat. They have some curvature, but they are pretty flat. And I think that makes it very easy for M1 to control the plant. The job of M1 is to control a plant of bones and muscles. And it's much easier to adapt and effect more linear control than non-linear control. So my hypothesis is that as you go from early stages of sensory encoding to motor control, you gradually linearize the representations, and by the time you get to M1, you get away with linear methods.
So I also have a question then. So I'm impressed by the power of linear methods, and also you showed us in another talk that these manifolds, you can learn from the manifolds even across different individual of the same species,
right,
and maybe or even across at least primates. And might be, and from what I see, you should think more a circuit guy. So circuit-wise, I think that these manifolds somehow embeds the possible activities constrained by the connectivity.
Right.
Okay. I hope.
Right.
Thanks. And so I got it. And but then this maybe means that not all since we can reduce the dimensionality, not all the details of the connectivity matter after all. So the most of the intricacy of the Connectome project, this thing of tracking every single dendrite, maybe it's due to biological constraints and it's not functional.
In the primate brain, it cannot be important that neuron 17 talks to neuron 34.
Yeah.
What has to be important are statistical patterns of connectivity. And our intuition is indeed that what makes a system have a low-dimensional dynamics is the connectivity. And the question is which features of the connectivity are important. And there is a beautiful set of papers coming from Zoltan Ostojic at the Conor Mann in Paris, where he shows that if your connectivity matrix is low rank, so it looks full, but it's actually obtained from the outer product of a pair of vectors or two pairs of vectors, then the resulting dynamics is low dimensional. So it's a sufficient condition, it's not a necessary condition. Maybe there are other ways of making the dynamics low dimensional, but that's an example, and it follows precisely into what you were saying. I don't care about the details of the connectivity. I only care that it's low rank. If it's low rank, it's sufficient.
Thanks a lot. If there are no other questions, let's thank the speaker again.